All right. So let's kind of take a look on the lab number 5, fungi and lichens. And you can see we already discussed the sexual and asexual spores. In this case, the structure which producing spores known as the sporangia. And you can see there will be asexual sporangia and sexual sporangia. No matter what, sexual or asexual sporangia, they will produce haploid spores. Now, with the sexual sporangia, what will happen? We'll have a meiosis taking place, right? You see right there, meiosis. And in this particular case, since it's a meiosis, it will produce genetically different spores. So those spores, also known as the myospores, would be genetically different. Even though both of them are produced by the sporangia, the structures looks almost identical. The only difference is where they're growing from, so to speak. All right. So here we'll talk about the table number 1 momentarily. Pre-lab questions: are the fungi prokaryotic or eukaryotic? They would be eukaryotic organisms. They one of the kingdoms belong to the eukaryota domain. Heterotrophic, autotrophic, something else. They are exclusively heterotrophic. They have to have some sort of food in form of the organic compounds. What supergroup fungi belongs to? It's called Apistoconta. I think it should be somewhere here, right? Yeah, here we go. Apistoconta, the same supergroup we, not humans, well, humans as a part of the animal kingdom belong to. So Apistoconta, are spores haploid or diploid? Please remember they are haploid, okay? Meaning they will be a single N, not a double number of chromosome, but whatever the number of chromosome, a single number of chromosome for them. This is the normal for the fungi. For animals, 2N is a diploid number, is the normal for animals, but they are not animals, they are fungi. What 2 types of organisms make up lichens? Fungi, algae, 2 of them. Different types of the fungus and different types of the algae, creating different colors of the lichens as well as different species. What benefit do fungi provide to the other part of lichen? Meaning, what does it do for the algae? To the algae, well, first of all, fungi provide them with the protection, structural support. Remember, they have the cell walls made out of chitin, which is fairly rigid. Also, they provide them with water and minerals. All right, now we're looking at the lab number 1. This is how the table number 1 will look. You will have the model as well as the microscope, and we'll talk about that momentarily. So first, mycelium. Is it haploid or diploid? Mycelium, all of those strands in fungi, they are almost all haploid. Now you will need to label. Make sure you're labeling, right? Labeling is relatively easy. Whenever you're labeling, it's a really good idea to kind of keep mycelium right here, so it's not like all the spider web. Some people are just using the arrows, some people are marking it right there. That's fine, as long as you're doing it correctly and I can read it, that's fine. So mycelium, those particular, any of those strands are all of that, all of them are mycelium. So the whole thing is mycelium, okay? A specific strand is a hypha, plural hyphae. Now, zygosporangium. Zygosporangium have to go from a zygote. Where the zygote? This is where 2 strands are meeting and they creating the zygote. So you'll see that particular brown structure. So the sporangium, the spore-bearing structure have to grow from the zygote. So that one would be a zygosporangium. Now, asexual sporangium. Asexual sporangium will grow straight out of the hyphae. There is no meeting each other. no exchanging of the genes, no meiosis, nothing. So in this particular case, that would be asexual sporangium. Now it's talking about sexual sporangium. Sexual sporangium, also known as the zygosporangium. Therefore, this one would be the structure for the zygosporangium or sexual sporangium. Make sense? All right, moving on. Note that two sporangia can be moved or swapped on the model. You literally can pull them out and swap. Which is more important in telling the difference between sexual and asexual sporangium? The shape or where it's attached. Where it's attached. If it's growing out of the zygote, it is a zygote, aka sexual sporangium. If it grows straight out of the hyphae, that's asexual sporangium. Both of them will be producing spores. That one will be producing myospores, right? But the spores are haploid, of course. So, and in explaining your answer, I just did. Basically, if it grows out of the zygote, a diploid structure, it will produce the sex spores, sexual spores. And if it's growing straight out of hyphae, it's asexual sporangium. So this is essentially what we see happening with the sexual reproduction. The hyphae from a positive, hyphae from a negative strand. Could be either, it could be negative, it could be positive, but they start growing toward each other. And here they are like, hey, hello. So that's basically dating, showing dating in the plants. Like, hey, you positive, hey, negative. And then now you see those particular two cells, they are separating, they are next to each other. And then they merge. That's the reason why there is no males or females in this particular case, because both of them contributing, both of them donating, and also those cells are accepting the DNA of each other. So this is like a dikaryotic stage. This is where we have two nuclei and then they merging. So now we have the zygote, and that particular out of the zygosporangium will start growing. So zygosporangium, this is particular structure which will become a zygosporangium. Zygosporangium haploid or diploid. In this particular case, it's a diploid structure, that middle part. That's essentially a fungal baby, a mold baby. Cute, right? So in this case, we're looking at that structure. So will it form a sexual, asexual, or sexual sporangiospore? Because it's a diploid, it's a baby, it will have to go through the meiosis to become a haploid, and therefore it will be a sexual sporangia, sporangium, or sometimes referred to as a sporangiophore. It's the same thing as a sporangium. All right, now the slides. Also on that particular slide, well, here we're looking at the model, and again, look, here we have a sporangium growing straight out of the hyphae. That one is asexual. Now we're looking at the zygote and the sporangium growing out of diploid structure, producing haploid spores, but that in this particular case, we're looking at the sexual or zygosporangium because it's creating a zygote, right? How can you tell the sexual or asexual structure? Depending where it's growing from. If it grows straight from the hyphae, it's asexual. If it's growing from the zygote, it's a sexual, and momentarily I'll show you the pictures. So in this particular case, hyphae, is it haploid or diploid? Since it's growing from the haploid spore, the hyphae are haploid. All right, let's take a look. Yes, here we have the zygosporangium. Here we have those particular models for your reference only. Once again, do not copy and paste already labeled picture whenever it's asking you to label it. I had students before doing that. Please don't. Not the main idea behind that. So now let's take a look on that particular picture. Actually, that one. Here we have one hyphae, another hyphae creating the zygote. So is it sexual or asexual reproduction? In this case, since it's creating that brown thingy, the actual picture, actual slide of the fungal baby. Here you can clearly see that we have one hyphae, another hyphae merging, actually two of them right there. So here we're looking at the zygote. If it's a zygote, it's a sexual reproduction. Okay. So this is where this particular picture can go, and you will need to label hyphae, that particular kind of greenish, tealish color, strands, all of that are hyphae, right? And the brownish structure are the zygosporangium. I guess I flipped them. Let me change it then. So anyway, now here you're looking at the sporangium, but it grows straight out of the hyphae. So this is where it goes. This is where you need to look it up while putting this one. So again, there is no meeting, no zygote, nothing. It's just the tons of sporangia coming straight out of the hyphae. That's asexual reproduction. Really, really fast, especially if plenty of food. Don't have to meet anybody, don't have to worry about the zygote, don't have to do anything. Just sprung the sporangium straight out of the body and produce as many identical spore as possible. So hyphae haploid, of course, and this is the asexual structure in this particular case. Why it's an asexual structure? Because it's growing straight out of the hyphae. And then we'll already talk about those ones. So is a sexual or asexual reproduction? This is a sexual reproduction. Is hyphae haploid? Hyphae is still haploid, but the structure, that one right there, the zygote, it's actually a diploid structure. And then out of the zygote, what we'll see, the zygosporangium growing, right? That particular zygosporangium is what is the structure would soon be here, zygosporangium, this thing. So we're looking at this thing, and then later on, we'll see from the zygote, the zygosporangium growing. Make sense? Any questions, any issues? All right. Moving on. So make sure you will be able to tell what it is, haploid, diploid, which part is haploid, which part is diploid. The light one is haploid, the brown one is diploid. Which one is the sexual, which one is asexual sporangium. If it grows straight out of hyphae, that's the asexual. You can actually see on that part two hyphae meeting with each other. Eventually it will will create the zygote, and then the zygosporangium will start coming out. All right, so Basidiomycota, next one, next group. Yeah, showing you this. Yes, here we go, some of the pictures now. Basidiomycota, quite a few. The shelf mushrooms, you probably know what they are. You've seen it on they quite often grows on the decaying trees. They look like tiny little bitty shelves. So in this case, what we're looking at is the actual mushroom part. This is the part which producing spores. We have a quite large, this is a shelf mushroom right there, but that particular shelf mushroom is large. I mean, it's big. And if you pick it up, and it's been there from the beginning, I started, and that happened quite a few years ago. And if you'll take that particular shelf mushroom and shake it, you will see a tiny little bitty spores still there coming out. So anyway, preserved mushroom, basidiocarps type, cap and gills. Well, let's take a look. Here we have the preserved mushroom, dried up mushroom. The whole mushroom thingy right here, that's the basidiocarp. Okay. Now cap, everybody can see the cap. Inside of the cap on the back side, you will see the lines. This is known as the gills, and that stem-like structure is the stipe. All right. We already talked about this type when we were looking at the stem-like structures in the algae. It's not a really stem yet, but it's a stem-like structure. So here we have a relatively large shelf So here we're looking at the cap. The whole thing is the basidiocarp. Here on top, we're looking at the cap, this type, and here we start seeing the gills. You see those particular kind of lines. Why the lines? Remember, this is the structure which produces spores. So more basidia you can fit in one mushroom, more spores you can produce. So it's drastically increasing the production of spores. They're producing billions of spores. So here we're looking at the cap, a back side of the cap with the gills and this type. The whole thing is the basidiocarp, the mushroom itself, right? So on a preserved specimen, where would you look to find the basidia? In the gills. So here we're looking at the shelf mushroom, and that's a general picture of that, showing you the cap, the stipe, the gills, and the whole thing, the mushroom itself, is the basidiocarp. All right. So again, kind of showing you the basidiocarp, gills, and within the gill, you can see this is the cross section, and this is the basidia, the club-looking structure with the spores. The spores would be a myospores, right? Myospores means the sexual reproduced structures. Now we're looking at some of the examples, and you can see spores right here. Now look, what are you looking at in this particular case? You're looking at the cap with this type, which has been cross-sected. So this is part right here, the outer part is actually the stipe. This is the outer part of the cap, and this is the gills. And the gills have tiny little bitty brown dots. All of those brown dots are the spores. All right. So here we're looking at the stipe. One of those lines is the gill, and this is the outer part of the cap. All right. Now, if you look under the closer magnification, you can still see a little bit of stipe, and now you can start seeing the gills. But then, whenever you're increasing magnification, you can see the gills, which are dotted with the spores. Now, let's kind of look at the higher magnification. Look, this is the part of the gill. Here, at this particular teal-looking structure, is the basidia, a single basidia. Or right there is another basidia. On top of it, you will have, usually you will have 4 spores. But you can see, well, 1, 2, 3, 4. On that basidia, you can see 1, 2, and 3 spores. Why 3? One of them already probably fell off. Here is another basidia, and it has only 2 spores. 2 others just, it's either are they behind or they already fall off. So, but that particular teal-looking structure is the basidia. The brown-looking structures are spores, and spores are, of course, haploid. So let's take a look. Stipe, gill, basidium, basidiospores. We just look at it. Now, if you look at the higher magnification, you will be able to see the spores and the tiny little bitty structures where the spores are growing. This is where meiosis actually occurs, and this is why we have 4 spores. Which part of the slide has the characteristics of the club shape? That particular gives the name stipe, gills, basidium, or basidiospore. The basidium. That basidium means club. Okay, it's Latin. Basidium, club. So, and then the spores which are growing on the basidia, known as the basidiospores. So, club shape is the basidium. Are the basidiospores haploid or diploid? Spores, haploid. Is the example of sexual or asexual reproduction? Since it says basidio, and we know in basidio meiosis taking place, it would be sexual reproduction. Any questions on that? Any problems of identifying any of the structures? And again, I would highly recommend you to, well, if you can sketch, that's wonderful. If not, just print screen it, or there is a snippet thingy or whatever it is, and I'll put the picture from the guide. Or once again, you can use internet. I don't care if you're using my own pictures, but with my own pictures, I already gave you the permission to use them. If you're using it from the internet, you have to have a permission from that particular source. Like let's say you'll go in the McGraw-Hill and we'll take it from McGraw-Hill. You'll have to have it's copyrighted by the McGraw-Hill. So you'll have to have the permission from the McGraw-Hill, or look for something which is known as a Creative Commons. That's a license which allows you to use those images for education, not commercially, but for the education. But then you will need to create the reference at the end of the lab. Okay, either or, I don't mind if you're using somebody else's work. If it's your own, be my guest, but make sure you will say that it's your own. So next table, say that you'll see this type, and again, it's a cross section. We have the cap, and then it has been cross-sected. So stipe, gills, and then the basidium. All right, now here we have the Earth star, because whenever it's first, it starts as the ball, and then whenever the spores are ready, it opens up so the spores can be picked up by the wind. And this is the morel, and again, we're looking at this type, and we have a cap of the mushroom. And now we're looking at the cross section with the ascus. That's a Marcella. Marcella is a species of the Ascomycota. Now we start looking at the inner side. This is the cap, and this is where the ascus will be. Well, this is all ascus. So here we're looking at the sac-looking structure, right, which is filled up with the purplish bluish dots. Those dots are myospores, or specifically because they are located in the ascus, they are ascospores. So sex reproduced spores. And in this case, we have 1, 2, 3, 4, 5, 6. We're supposed to have 8. Where are the rest of them? They might hide behind, or they might be already released, a part of them. So in this particular case, you're taking this picture and label ascus, the whole entire structure filled up with the spores. That's the ascus, which means sac from Latin. Hence, that's the reason why they call it sac fungi, because under the microscope, it looks like a sac filled up with the spores. And then the ascospore, one of those dots or round structures are ascospores. Ascospore, haploid or diploid? Haploid. What are the similarities between the ascocarp and basidiocarp? Both of them has stipes and the caps, right? Because it's exactly the same. It's a convergent evolution. They start look alike since they need the same function to take the spores out of the ground into the air and disperse them. What difference between two of those type of the fungi? Well, there is several major differences. One of them, Ascomycota will produce 8 spores per ascus, while the Basidiocarp will produce 4 spores per basidia. And then they sticking out of the basidia, 4 of them. And here you have 8 inside of the sac, so to speak, inside of the ascus. What difference do you see in the arrangement of ascospores and basidiospores? 4 basidiospores, and they kind of in the open. 8 ascospores, and they are closed in a structure known as the ascus. How are the ascospores and basidiospores similar? Well, both of them haploid, and both of them are produced by meiosis. They are sex spores. Any questions, any problems there? Any issues on what you're looking at? See? Showing you the ascus, the whole entire structure, and the ascospores. Yeah, the picture is a little bit different, but this is what you actually would see if you'll go to the face-to-face lab in our class. I took it literally in our class, in our lab. Now, table 4, known as the Deuteromycota. Deuteromycota is not the actual phylum or division. Division and phylum is pretty much the same thing. It's the same level of the classification. It just division usually referred for the plants and phylum for the animals. So I'm a zoologist. I have a zoology background. Remember my favorite species, right? So I generally have a tendency to say phylum for the fungi as well as the plants. And some of the botanists like, it's not a phylum, it's a division. Well, it doesn't really matter. I don't want to overcomplicate. And on this particular level of biology, just introductory biology, I think it's not extremely important if it's a division or is it phylum, if it's a botany, if it's a zoology. And again, it's my zoology background which tells me to call it all phylum. If you decide to go with a division, that's fine as well. As long as you're not calling it a division for the animals, I'm fine. If you do, I'm like, that should be phylum, but I would not count it as a huge error. I might write you a little note about that, but other than that, anyway, going back to the Deuteromycota. If you can clearly say this is the ascospores, so that have to be Ascomycota. This is a Basidiospores, have to be a Basidiomycota. The imperfect fungi, we do not observe, for one or another reason, the sexual reproduction of those particular creatures. Therefore, we cannot say are they Ascomycota, Basidiomycota, or Zygomycota. Okay. In many cases, we think that it is they in a rare occasion they engage but into asexual reproduction, but they are generally reproduced asexually. Hence, the name imperfect fungi. Basidiomycota could be there, Ascomycota could be there, Zygomycota, as long as they are reproducing only asexually. So, and the asexual, of course, they will produce sporangia, also known as a conidiophores. A penicillin, for example, is the Deuteromycota. Remember the nasty Aspergillus fungus? Yes, it's a Deuteromycota as well. We not really sure how they do it. I guess they shy to show up, or they just completely lost the ability of sexual reproduction. So here we're looking at the Aspergillus, common, very common mold. I'm telling you, you're looking outside, there is tons of Aspergillus spores flying around. Most likely we will have it in our house. So it's a very, very common, but again, it's the opportunistic. If it will find the host which is not really have a really good immune system, then it might start invading. But other than that, not a problem. So this particular greenish bluish thing is so-called the mycelium. That particular thing is mycelium. All of that is still a mycelium. The dots, most likely, the black dots, most likely are the spores. Now here we're looking at the sporangia, right? There is a conidiophores. Here they call conidiophores, and they will contain tiny little bitty spores, which known as the conidiospores. Everything else, well, that's the black structures, and everything else, the bluish tealish structure, that's the mycelium. So in this particular case, is it example of sexual or asexual reproduction? We just talk about the Deuteromycota, known for the asexual reproduction. What's the benefit of asexual reproduction? Quick, really fast. You don't have to meet another one, figure it out if it's a positive or negative. As long as you have a substrate, you can grow really fast and produce as many babies as possible. Oh, spores. What is the benefit of sexual reproduction? genetic variance. We already discussed that, I already told you. But in this case, genetic variance, genetic variety. The spores become genetically different from each other. As a result, it helps the organisms, any kind of organism, in this particular case, fungi, to adapt to any kind of environment. All right. So here we're looking at the conidiophore and the spores all around. On the background, those thread-looking structures or stick-looking structures, that's the mycelium or part of the mycelium hyphae. Now, penicillin, here we have some more of that. On the penicillin, I was unable to find a really good slide, so I didn't take the picture. Therefore, it most likely will not be on your lab practical. That will not be a really good thing for me to give you, but it will look like a nice looking tree, like a palm tree with the dots coming out right there. I don't know, I could not find a good sample of the penicillin. So, but essentially it looks very similar, just structure of the conidiophore a little bit different. This is what you saw right here, right? Yeah, I think this is the picture they were using. And for the penicillin, we don't really have it, but you can take, it will look very similar to what you see. In this particular case, you can just, or you can find the picture on the internet, but that picture will do. I'll give you the permission to use already labeled picture. And conidiophore, conidiospores, mycelium, all of those strands are mycelium, conidiophore, conidiospores. Why do you think it would be helpful for the fungus to produce substance, a toxin, penicillin that kills bacteria? Competition. It's competing against the bacteria for food, so it needs to show off. What about us? Can the penicillin kill you or me or anybody in humans? Yes, but at much higher concentrations. It was designed by that particular fungus to destroy its closest competitors, which are the bacteria. They are really, really good in killing those. All right. Any questions, any problems right there? Now, lichens, we talk about the three major forms, the crustose, foliose, and fruticose. So kind of crusty looking one. That particular white stuff looking like a crust, and the orange look like a crust. That's two different types of the, two different species of the lichens on that particular twig. So you will need to draw the example. So again, you see the orange ones, the white ones. That's the crusty fruticose. Here we're looking at the leafy one, foliose. And again, it's on the twig. Is it parasitizing? No, it just needs the place to live. So here is a piece of branch which I found on my backyard, and I brought it to the lab. So, but you can see a really nice leafy looking lichens. Lichens are actually very, very sensitive to some of the air pollutants, such as phosphates as well as the sulfur oxides. And if you have, it's basically an indicator that you have a clean air. If you live in the area where you can see lichens on the trees or on the fence or anything like that, it means that the air is fairly clean. So in this particular case, foliose, right? Leafy one. And here we're looking at the shrubby one. Looks like some sort of alien Martian plant, so to speak. And you can see same twig. Actually, I was cleaning up the backyard and I pick it up like, example for the Bio2 class. So you're looking at those particular shrubby ones. Looks like a shrub, but in this case, you're looking at the shrubby one and you're looking at the foliose at the same time. Two different species of the lichen. Here we have a leafy one and the shrubby one, right? Lichens are made out of what type? Two types of the organisms, the fungi and the algae. All right. If a lichen left too long in the dark, what's going to happen? Well, the fungus by itself does not really care if it's a light or dark, inside, outside, don't care whatsoever. But the algae, in order to have a photosynthesis, carbon dioxide, water, and the light. Remember, don't forget the light whenever you're writing that particular reaction during the test. Don't forget to write light, because without the light, photosynthesis is not happening. Light, photo in photosynthesis means light. Light synthesis. No light, no synthesis. Synthesis, of course, is a combination of carbon dioxide and water into something sweet, glucose. So if there is no photo, if there is no light, photos means light in Greek, right? So if there is no light, there is no synthesis. So make sure you understand that. So in this particular case, if you leave it too long in the dark, no light, no photosynthesis, photosynthesis not happening, the algae will start starving out and dying off. As the algae are dying off, the fungus will start dying off, because algae are primary for, well, they are food source for the fungus. They're not eating the algae, algae is just sharing the sugar. So in this particular case, the lichen would slowly die. Do you think lichens harm the organisms they grow on? For example, in this case, did they harm that particular twig or that particular branch of the tree? No, they did not. As a matter of fact, they were living on the dead part of the tree. So in this particular case, they are self-sufficient. They're not parasitizing. Parasites usually using the host. Remember, one is benefiting, another is not. In this case, it's example of the commensalism. The plant doesn't mind that the lichen lives on it. Within the lichen, you have a mutualistic relationship. Algae gives something to the fungus, and fungus gives something to the algae. All right, and I think, yes, here we have some pictures, and we are done with the guide. So post-lab review questions. Which phylum of fungi? Sac. Sac means ascus. So Ascomycota. Mycotus again means fungus. Fungus with the sacs. Sac fungi. Which phylum does black mold belong to? Most of the molds, not all, but most of the molds belong to the Zygomycota. What type of structure grows out of zygosporangium? Spores. Zygosporangium will produce spores. What is the type of reproduction that phylum Deuteromycota is known to use? Asexual reproduction only. Which type of lichens forms a big leafy structures? That would be, of course, foliose right here, right? In lichens, what part contributes the food in the relationship? Algae, through the process of photosynthesis. What is the mycelium? Mycelium is overall the mass of the hyphae. What is the function of the stipe in the Basidium, in the phylum Basidiomycota? Stipe takes the cap out of the ground, and the cap, this is where we have the gills, this is where the spores are produced, so spores can be distributed through the air or wind, by the wind. Name 2 species of fungi, they produce conidiospores. Conidiospores are Deuteromycota, and we already talked about 2 of those. That's the Aspergillus and the penicillin. What type of fungus is Marcella? And we just did that. Let's see. Whoops. Marcella, right? This guy right here, a girl, whatever. Positive, negative. So in this particular case, it belongs to the Ascomycota. It's a sac fungi. All right. Now, penicillin belong to a Deuteromycota, as well as Aspergillus. So 2 of those, Deuteromycota. Caprinus, here we go. That's the Basidiomycota. Rhizopus, black mold or mold. The Zygomycota. Marcella, we just talked about that. Here we go. That's the Ascomycota. Now, spore-bearing structures. For the Deuteromycota, it would be conidiophores. We talk about that right in here. Conidiophore. So that would be good for the penicillin, conidiophore, as well as aspergillus, conidiophore. Caprinus belong to the Basidiomycota. So spore-bearing structures would be basidia. For the Rhizopus, belong to the Zygomycota, Zygosporangium. And for the Ascomycota, Ascus or Asca, plural. Now, the name of the spores will come from the spore-bearing structures. So if this one is a conidiophore, the spores will be called conidiospores. The spores which has been produced on the conidias. Make sense? Now, Basidiomycota, on the Basidio, the spores would be Basidiospores. Aspergillus, Deuteromycota, conidiophores, conidiospores. Rhizopus, Zygomycota, Zygosporangium, zygospores. Last but not least, Ascomycota, Ascus, ascospores. The spores are haploid, right? Any kind of, any of those spores, all of them will be haploid. Penicillin and aspergillus, that would be asexual spores. Everything else are sexual reproduced spores, also known as the myospores. So here we have a myospore. That one is a myospore. That one is a myospore. And your favorite reflection questions. Thank you very much, guys. I'm reading right now. I was grading a couple of labs before the class started. I was looking at those. You are doing a wonderful job. Thank you for those short paragraphs. Gives me the feedback from the students, since we don't really have much of the feedback in the lab. It's much easier in the face-to-face lab to see the feedback from the students, see what they learned, what they didn't learn, and can they apply it in the real world, so to speak. That's the reason why I'm keeping it there. All right. And we are done for today. Thank you very much for stopping by. Hopefully I'll see you on Thursday.